So I feel like I'm seeing a lot of friends here, and I really love the chance to talk. But I've been given an impossible task today because I remember being with Elizabeth and Pat here 6 or 7 years ago and talking about setting up STAR. And since that time, as Pat mentioned, there's active projects in CMT1A, CMT1B, CMTX, CMT2A, CMT2E. There's plans in place for the recessive forms of CMT. There's partnerships with multiple companies. And if I talk about all of those, I'll go over and Elizabeth will yell at me. And in addition, I want to do a second part of my talk because none of this works without partnership with you guys, and not just by allowing us to evaluate you, but actually in necessary partnership to take these compounds to patients. So what I'm going to do is I'm going to talk briefly about STAR projects for 3 of the most common forms of CMT. Carly talked about, that's going to be CMT1A, CMT1B, and CMT2A, and then I'm going to show you how we take that information to use it in future trials. So several people have talked about this already. There's more than 80 genes that cause CMT now, and these are scattered throughout all the chromosomes. And again, to try to make sense of these is a challenge, but it also allows people to understand the biology of how demyelination works and axonal degeneration works, so that when treatments are developed, they're based on reason and science. And again, John's introduction was really helpful for this. And because of this, and Pat talked about this as well, the CMTA decided to take the fact that there is a scientific basis to try to develop treatments, and rather than just receive grants and not know how they were going to be focused, to actually bring together a group of people who really were experts in the field of the biology behind CMT and try to figure out what was necessary to do to bring rational treatments that could be effective to patients, and to go anywhere in the world and pick the best people to do the projects, and that way to try to sort of exponentially increase the speed by which treatments came, and rather than taking a more passive approach. And I'm going to show you this in these 3 disorders, but I think that the model that Pat laid out is worth setting out again just at the start, so to see how these 3 diseases are moving in this model. So the first step is to figure out what there is that you can measure in an assay to look at large numbers of potential medications that might already be available in pharmaceutical libraries or at NIH libraries, and just figure out a way to screen to see if any of these compounds can be effective in these diseases in a quick screen model. Then to take hits that come out of that quick screen model and look at them in a little bit more specific way. First, in other types of cells, and maybe even including human stem cells, which I'll show you a little bit about, and then into animals, but to make sure the animals aren't done with too few numbers and aren't done by people who have a bias to have their compound work, so to put it in the hands of neutral experts like the Psychogenics facility, and then take them into clinical trials. So I'm just going to show you 3 examples of that, and again, so much has happened that I can't go through everything in all of these diseases. So I'm going to start with the most common form of CMT, which is called CMT1A, and it's caused by a duplication on chromosome 17, and this duplication contains a protein that's named based on where it is. So it's in peripheral myelin, and it's a protein, so it's called peripheral myelin protein, or PMP, and its size is 22 what are called kilodaltons, so it's called PMP22. And as Carly has pointed out very nicely, if you have a duplication of this, you get too much PMP22, and that causes CMT1A. And there's other families who get too little of it, and because they have a deletion instead of a duplication, and these families have something which has, I think, the worst name ever invented. It took me a year to learn how to say it, but it's hereditary neuropathy with liability to develop pressure palsies, so HNPP. And this is just a skin biopsy showing nerves going up under the skin up to pressure sensors in the skin. And that's just to show that in a non-invasive way, and again, Pat talked about this, you can look at nerves, and you can do a technique called immuno-electron microscopy, and you can use little particles of gold to label to antibodies, and you can then count how much protein there is in the myelin. And this is a busy slide, but I'm just going to show you with this arrow that in people with CMT1A, they have a lot of grains. Each of these dots is from a patient. Control, or people who without CMT1A have less, and people with HNPP have still less. So the strategy then is to figure out ways to try to regulate the amount of PMP22, because in the end, it's not that each PMP22 gene that you're inheriting has a mutation in it, it's just that you get an extra copy, so there's too much of it. And again, in the interest of time, I'm not going to spend too much details on this, but the initial question for the STAR group was what to use to screen for compounds to enable us to be able to look at large lists of medications. And the answer was in cells, because if you have a cellular model, you can look at thousands and thousands of compounds in a single day. And I always like to ask difficult questions. So if you look at these cells, you'll see, what color are they? And I'll give you a clue, they're green or blue. Not maize or blue, for Carly. And that's a key to this, because if you're looking for a signal in cells, it's very hard to measure the amount of a protein like PMP22. But if you could make a marker and put that in the cell so that they turn green or blue, it would make things much easier. And that's behind the strategy that the STAR team used. And I've heard many kind things said to me over the last 2 days, and also to John and the team at Stanford. But I just have to give a shout out to this person here on the top. This is John Severin, and he doesn't see patients, but really, in many ways, he drives the science behind all of these projects. And because of John's work, in large part, I think we're in exciting times for all the diseases I'm going to talk about. But we're really here just a lot because of John, and I just hope the word gets out of what a great person he's been to the CMTA initiative. And what John did was he developed the cell lines that we now use in the screens for CMT1A. And this is my technical bit here. So as I say, this in scientific terms is what we call a straight line. Okay? And if you look at these boxes on here, these little boxes are the exons, the parts that code a protein, the pieces of the PMP22 gene that make a protein. And Carly just talked about whole exome sequencing. These are the parts of the gene that get sequenced in exome sequencing. But in between exon 3 and exon 4, there's a piece of the straight line with no boxes on it. And that's called an intron. And the intron doesn't code for the protein, but it has effects on the amount of the protein that gets made. And in this area here, John Severin actually was the scientist who discovered that the DNA sequence in this area between exon 3 and exon 4 is really the driver of PMP22 expression in the peripheral nervous system. It has a couple of sites which are called, named, and they're called what are called transcription factors. One is EGR2, which, by the way, is the cause of CMT type 1D, and SOX10, which is a cause of another form of CMT. But those two transcription factors regulate the transcription or the levels of PMP22. And so what John did was he took in genetic engineering, he took that intronic sequence out and he stuck in another part of DNA, another couple of exons, which make something called firefly luciferase. And so what do fireflies do at night? They glow. Thanks, I need help here. So they glow because of luciferase. So if you take the gene for firefly luciferase and you put that in these cells, when PMP22 gets turned on, the cells glow. And he wanted to have another way of measuring it to make sure there wasn't just some artifact from the luciferase, so he put an enzyme called beta-lactamase in, which is this blue box. And then he took these constructs and he put them in a cell line, which are called S16 cells. And I'm not going to spend a lot of time with that, except to say that they regularly make PMP22 or myelin proteins, so they're a good model for Schwann cells. So we have all these cells. The question is what to do with them. And many of you may have met Brittany Wright out there, who has a nice poster on similar work with CMT1B. But what I'm going to show you is the system that Brittany uses and that Jim and Glacy at the NCATS facility use to test the cells that John Severin put together. And this is a busy slide, but it just goes through the model that Pat introduced again. So compounds get taken through these screens on the left here, and then as they move down, they get more and more specific towards PMP22. So for hits from the original screens then get actually looked at to see how much PMP22 they're making. And then finally they go into animal models, which I'll show you in the next slide. And then they hopefully will go to clinical trials. And this is just an example of a compound which came out of this, which with positive results, but which is neurotoxic so that we can't use this in people. So here on what we call the x-axis going along here are the different concentrations of the medication that we use in the assays. And what you're looking for is for the level of the firefly luciferase to decrease as you get in the concentrations. I'm sorry, my mouse stopped working here. There it is. So you can see for the firefly luciferase, as you add more bortezomib, the brightness drops. The same is true for the beta-lactamase, but for measures of toxicity for the cell, that doesn't drop. So again, this is a positive result, and this is what we look for in the screens. I'm just going to ignore the bottom one here because it doesn't add to the story. But what I want to show next is what Pat and Mark Schuyler and the other members of the CMTA STAR team did, which was they went out and found the best animal models for CMT1A, which is a rat made by Klaus Nave, again going all over the world, in Göttingen, Germany, and a C3 mouse. We went to the Netherlands to get this animal from Frank Bass. And now these animals are in New Jersey, near the Tappan Zee Bridge, in a facility where it's paid for by the CMTA. They're housed there, and anytime compounds come through that screen I just showed you in studies that are not biased by investigators. Psychogenics has no skin in the game here. They look at the candidate medications and they test the animals to see if they'll be effective in animals, and then they will go into clinical trials. So that's the CMT1A model. Here is another one, which Brittany has her poster out. And again, in the interest of time, I'm not going to spend too much time talking about this, but here in CMT1B, this is caused by mutations in a protein called myelin protein 0, which is the major protein in myelin, and there's over 200 different mutations in the MPZ gene which cause the disease. So here it's not a duplication or a deletion. It's not based on dosage. It's based on a problem caused by the mutations in the protein. The protein does something it's not supposed to do, and we call that a gain of function. It's like in my household, I'm always doing things I'm not supposed to do. So my wife thinks of me as a toxic gain of function. And it's the same thing with this protein here. And the way many of the cases of CMT1B cause a toxic gain of function, maybe up to 40% of these mutations, is that they get stuck in the middle of the cell. They can't get out to the myelin. They get retained in part of the cell called the endoplasmic reticulum. So when a gene gets turned on in the nucleus of a cell, the DNA gets turned into RNA. RNA goes out into the out into the ribosomes, and it gets turned into a protein, and then the protein goes into the endoplasmic reticulum to get folded. And in these cases, it can't get folded right. And this is a cartoon of that. So this here on the top is the endoplasmic reticulum, and then this is the nucleus of the cell down here. And on the edge of the endoplasmic reticulum are proteins that sit there. And when proteins can't get folded correctly, they send signals down to the nucleus to tell the nucleus to change its behavior to try to alleviate the process. And this is called the unfolded protein process, or the unfolded protein response, the UPR. And abnormalities in the UPR cause many cases of CMT1B. And I'm just going to show you quickly. The science can get deep, and I'm going to go fast, but I just want to make a couple of points. So the three arms of the UPR are something called ATF6, IRE1 alpha, and PERK. And each of these sends signals down to the nucleus. The ATF6, we're not involved with that much. That gets changed when it's active in what's called the Golgi apparatus, and that sends signals down to the nucleus. The one that we do use is called this IRE1 alpha, and when it gets activated, it turns this protein, it changes this gene XBP1, so it goes to the nucleus. And when the last one, the PERK pathway, gets activated, it activates something called CHOP, which goes to the nucleus. And so I'm going to show you how we use XBP1 going to the nucleus and CHOP going to the nucleus as markers of UPR activity in patients with CMT1B. And Brittany has a nice poster on this also out in the hall. So what we can do with cells is we can take engineering, as we talked about with John Severin on the CMT1A portion, and we can put a reporter in there. We can put a reporter on the XBP1 that only lights up when the XBP1 goes to the nucleus. And you can see that in this guy here. This R98C is one of the CMT1B mutations. And we can look in cells which have this mutation put in them and see how many green nuclei there are in the cells. And we can measure that with assays like with the Englacy lab. We also can look at skin biopsies again, and we can look for activation of these markers of the UPR. And here is a skin biopsy from control. I would say a normal control, but it's from me, so I don't know how normal that is. But this next one is from a patient who has CMT1B, and you can see all these pink spots here. And what that is that's that transcription factor CHOP going to the nucleus. So Brittany and people from my lab are working on putting these reporters into cells that will be able to screen in assays for CMT1B. And in the interest of time, I didn't put all the mice in this talk that are available, but there are mouse models for CMT1B. There's work on making stem cells for people from CMT1B. So the pipeline that Pat was talking about at the beginning is in place for CMT1B also as part of the STAR initiative. And the last example I'm going to show you just of the disease processes in the STAR initiative is CMT2A. As Carly mentioned, it's one of the 4 most common types of CMT. It's the most common form of CMT type 2, and it's caused by mutations in a gene that comes from the nucleus, but then goes into mitochondria, and mitochondria are the energy supplies for the cell. And to work well in nerves, individual mitochondria have to fuse together like grapes coming together to make a sausage. And in that state, they can go up onto the axon of a nerve, travel down, and provide energy to the cell. And one of the ways that they fuse is with a gene called mitofusin-2. And if there's mutations in mitofusin-2, that prevents that process and causes CMT2A. And here is what's called a Western blot, which shows the amount of mitofusin-1 in sensory nerve cells, which are called dorsal root ganglia, and as an example, in mouse embryonic fibroblasts, or MEF. And here's mitofusin-2. And what this slide is supposed to, or hopefully will show you, is that different cell types have different amounts of MFN1 or MFN2 in them. It's not the same in all cells. And work from David Chan, and then another rising star in the CMT world is Bob Balow down at Cedars-Sinai. And Bob Balow has done a lot of the work also, which shows that if you can increase MFN1, you can actually overcome consequences of the mutations of MFN2 in both rats and cells. And so, again, Pat has put together a team. We meet together, we talk about things on a monthly basis, and what the team decided to do was to take this information and look for medications which are out there in libraries from various companies and see if there are medications which can increase the amount of MFN1 to overcome MFN2. And this is another one of those slides with a straight line. And here it's just to show that the same type of reporters, the firefly luciferase, well, and here's another luciferase reporter, are put into cells linked to MFN1 expression to try to identify candidate medications that increase the level of MFN1 as a rational strategy to treat CMT2A. So I went through that pretty quickly, but I hope it was at least somewhat clear. Now, what Pat talked about was doing the cell assays in these diseases, getting the best people to do them, but also to recognize that mice and rats and cells aren't humans. So we needed to get a human assay into this screening process. And so that's where we are working hard to take skin biopsies or blood samples and turn them into immortalized pluripotential stem cells, and then turn those stem cells into neurons or Schwann cells to then be able to be used in screens. And this is just to show Mario's work with CMT2A. Mario was a postdoc in our consortium, and he took fibroblasts from, in this case, this particular mutation in CMT1A, a patient from that, turned them into stem cells, and then turned them into neurons. And I don't know if you can see this well on the screen, but the green on top is just to mark the axon. And this came from a skin cell, a fibroblast, just a skin cell, which was turned ultimately into a neuron. And these little dots below it in the same axon are mitochondria with a mitochondrial tracer. And what you can do is that you can then do time-lapse photography and you can see how fast the mitochondria move up and down the axons in these neurons, and you can do tests to look at the function of human-derived cells in 2A or in other types of CMT. And Pat mentioned we're banking fibroblasts from all different types of CMT to be able to take this process forward in multiple forms of CMT. Now, in this model system, once we got through the cells, we needed to have a preclinical animal model to test compounds in to see if they were effective before we actually gave them to humans. We needed organisms that have blood-brain barriers, blood-nerve barriers, kidneys, livers, to make sure that there's not toxicity, at least in other mammalian models, and we couldn't do that in CMT2A because there was no effective mouse model. There have been attempts to make mouse models, and people have overexpressed MFN2 and put mutated MFN2 on top of that and cause disease, but that's really not the same thing as the human situation. So with John Severin, again, leading the way and Bob Balo's help, a CMT2A rat was engineered with a knock-in mutation, exactly the way it occurs in people, and this was built and then sent to Psychogenics. And I don't want to take too much time here, but just to say that Psychogenics looks at these animals very carefully, and this is just an example of something called a NeuroCube, and these rats have abnormalities. This has just been found out. This is brand new results, which Pat mentioned in his talk. And we have another great member of our team, Steve Scherer from the University of Pennsylvania, who's the preeminent world neuropathologist for a CMT. And Steve's looked at these nerves pathologically, and these mice are clear models of, these rats are clear models of CMT2A. So we now have this system in place. So I hope I have another 10 minutes, Elizabeth, because you won't throw me off? Well, if you hadn't kept me waiting so much at the start. So the reason I want these extra 10 minutes is what I don't want to get lost is the role for people with CMT in this whole enterprise, because these projects are set up to generate good models and drugs, but we have to see if they work. And just a quick question, how can we tell if a medication works in humans with CMT? Sorry? Yeah, so if we gave people with CMT a drug, how would we know if it worked? I mean, I think one possibility is the one that John mentioned. We could knock you on your head and see if the CMT was gone, but I don't think that's likely to happen. I think the most likely scenario is that we'll make progress and get better and better medications. But what we have to do is we have to have ways to measure the effects in people, and that's why we developed, with help from the CMTA, the Inherited Neuropathy Consortium. History data, the outcome instruments, and the quality of life is what I want to talk about just briefly. So whenever somebody comes into one of our centers, including the excellent one here at Stanford, we do a CMT neuropathy score, which is a scoring system that we've developed over the last decade. We also have recognized that a lot of the changes in people with CMT occur in the first 2 decades of life. So with the help of Rick Finkel from Orlando, Florida, and Josh Burns from Sydney, Australia, we've developed a pediatric score, which we use. And I think in the interest of time, I'm just going to go through this quickly, but we take into account the age, sex of a person. We do hand dexterity measurements. This is a more functional score. We measure strength in a quantified way. We, again, measure sensation in ways that we can quantify. We have balance testing that's done on what's called this Brunins-Osaretsky test, which I can still not pronounce after 3 years. We do standardized motor function. And then what Josh developed, getting normal values, was an online calculator where you put all the values in that I showed you, including the person's age, sex, and you click the icon on the bottom right, and it spits back a score that's standardized to that person's age and sex during normal development. So now we have an instrument that we can measure change over time in children with CMT, which we're very excited about. These instruments I just showed you were designed from people like myself and the colleagues I mentioned. None of us have CMT, and we've sort of realized going along that it's presumptuous for us to think that we know what's important to people with CMT. So what we are working hard on in our outcome measures is to get input from people who have CMT. And these are called patient-reported outcome measures. And we have one which is Impress Now from Sindhu Ramchandran at the University of Michigan. And again, as a measurement of if medications are effective, we need to know how they affect quality of life. So David Herman from the University of Rochester has developed a CMT-specific quality of life instrument, which we have the first parts of that now also Impress. And then we were involved with clinical trials with CMT1A, and what it showed and emphasized was in how slow the progression can be using previous versions of the CMT neuropathy score. So it's become very important for us to develop markers of disease progression that we can detect change in over a year's time. And some exciting results come from our London center at the University College London, and that's with the use of MRIs in the legs of patients with CMT1A. And again, to get patient impact onto our patient-reported outcomes and our whole program here, we have what's called the Patient Contact Registry, which I invite everybody here to join if they're not already a part of it. You just have to go on RDCRN and Google that, and you'll get linked to the Rare Disease Clinical Research Network. We're the Inherited Neuropathy Consortium with a little cartoon of Charcot. And if you click on that, it will tell you how to link in and join the Patient Contact Registry. You don't have to provide any personal identification. And what happens then is we can send you questionnaires back and forth and just work together to really develop these patient-reported outcome measures. We keep track of the type of CMT that people have. So if there's any CMT subtype specific queries, we send them out to you. And again, this is really important for us. And this is just a distribution of patient contact registrants. We have about 2,000 patients around the world. And then the last thing, and then I'll shut up, Elizabeth, is that to really make sure we get everybody, we're also set up and are partnering with international, with an international registry that's housed at the same place as our Inherited Neuropathy Consortium registry is housed. And these are national registries from different countries around the world. We're trying to ensure that patients from all over the world, no matter where they're from, get evaluated the same way and are linked into all the efforts that Pat and Elizabeth and everybody have put together. So there's a million people to thank, and they're up on this slide, but really, I think going back to our meeting in Palo Alto a few years ago, just as something which really kicked off all of this, and I'm thrilled to be a part of it. I'm sorry I talked too long, and thank you all for coming.
Thank you.